when there is a need of it. When there is a need of it. Now, as soon as these points are exposed, it means now this antibody, which is biting on the antigen, it is activating its tail. It is willing to do some function. Here, what is the function? To bind the complement protein and activate. Now, of course, if there are many bacteria here, there is some injury is going on, it means there is some inflammatory reaction here, and if there is some inflammatory reaction in this area, it means that endothelial cells here there is some inflammatory reaction going on, and during the inflammatory reaction, blood vessels are dilated, endothelial cells are shrunken, and there is increased permeability. Again listen, because in this area bacterial invasion is there and bacteria are damaging this area, so naturally there will be inflammatory reaction, and due to that inflammatory reaction there will be vasodilatation as well as there will be increased permeability of microcirculation. So there will be more blood coming to this infected area, and there will be more proteins leaking out from vascular compartment into interstitial area of this tissue particularly. Because this becomes more permeable, so your sleeping dogs, who are your sleeping dogs? Complement proteins which are sleeping in the blood. Now they have a chance to come here so that they become active dogs. So what happens that from here different complement proteins, they will come out, right? But yet they are not active. These are not active. They are sleeping. Now what happened? Actually, by chance they will start hitting at this point. Whenever complement protein number 1, right, will hit over here, it will be activated. Is that right? Complement protein number 1 has 3 parts. Complement protein number 1 we can show like this, that this is complement protein number 1, right? And this has 3 areas, 3 legs. One leg is called Q, other is called R, third domain is called S. You know ABC, QRS, no need to be upset. And this QRS has nothing to do with the QRS complex of ECG. Is that right? So what happens, we say that complement 1 protein, right, it has 3 peptides or special domains QRS. This Q point can interact with activated antibody in complex with antigen. So what really happens, let's make it and see what really happens, that C1 will bind here, right, and when C1 will bind here, now you will tell me it has opened its eyes. It has become active protein or inactive? It has become active protein, right? And what are these points which are activating here? No, only Q is activating here. This is C1 which is activated. As soon as we come in touch with the antibody which is complexing with antigen, and which antibody? Not every antibody, especially IgG or IgM. Now it become active. When it become active, this is C1 activated, complement number 1. We put a bar. When we put a bar, it means it is active, right? So next time what happens? This is inactive complement protein, and this is active complement protein. So we can put a bar on it. So in the books when you find there is a complement protein written with bar, it means it is which form? Active form. So this is also active form. When it becomes active, it calls some other friends. Come here, there is an antigen antibody activity going, you are needed here. So its friends will come. What will be the next protein coming here? Oh, not directly C3. It comes C4. Right? Now, so second protein which comes over here, right, that binds here and become active. Is that right? Very happy. Because it is being activated by C1. So C1 activates the C4. It activates, this was C1. Okay, I will put it in black color. Or you want which color to be put here? Okay, this is C1. Now what is this? C4. Is that right? Why this protein is called C4? First of all, we should be thankful to the researchers that there are many complement protein, and we should be thankful and obliged to those researchers who did not put their names on these proteins. They just call it protein number 1, 2, 3, 4. And these numbers are assigned according to the sequence in which these proteins were discovered. So unfortunately, protein number 4 was discovered before the 2 and 3. So that is why it looks a bit out of order. Is that right? But this was the order in which they were discovered. Right. So complement 4 is activated by C1. Right. Once it is activated, normally, now I have to make another diagram here. Let's suppose this is an inactive complement protein. It's just like a baby. And normally it has a very special domain, and it is like a, yeah, what is it? It looks like, you have seen? It's apple, sure. So you can say this protein has two parts, a baby part and baby with apple. This complement protein has two parts, baby part, apple part. Or we can simply say baby for, B for baby, and A for apple. So it's B part and this is A part. So most of the complement proteins have A for apple, B for baby. Not difficult to remember. This A and B, when they are complex together, they are inactive. When they are stimulated, look, baby is sitting like this with an apple on its head, and it is activated, what will happen to apple? It will go away. So what we will say, that when it is activated, right, baby become active, right, but apple has gone away. What is left behind is, for example, complement, which fragment? Baby fragment, B fragment. And which fragment has gone away? Complement, A fragment. Is that right? So many of the complement, right, they consist of a complete protein, and when that protein is activated, some of the complement break down to two components. One component is called B component, other component is called A component. Is that right? Am I clear? B for baby, A for apple. But here B is not for baby. Here B is for the fragment which remain binding with the original complex. One fragment fall away, other remain bound. B for binding. Let's see what happened. This was C4. When C4 is activated, which component is lost? Yes, please. Right? So what will be this? C4A. And what will be this? C4B. So what we can say? That when complement number 1 is activated by the antibody, Fc portion of the antibody or tail of the antibody activate complement number 1, complement 1 protein activates the complement number 4, and complement number 4 hydrolyzes or breaks down into two components. One remain bound with this is called fragment B. One fragment diffuses away into fluid form, and that is called fragment A. So apple goes away, baby stays there. Am I clear? Now, once this baby C4B is there, it will attract another baby. And what is that baby? That also become active. And that is C2. And which fragment remain behind? B fragment. And which fragment will go away? Another apple will go away. This apple was from here, another apple is from here. What is this? C2A. Yes.
Did C1 also have complement A?
Of course not, as we can see there.
It just started.
Yeah, it never had apple. It never had apple, it's not going to lose it. So her question was that, will C1 break into A and B fragments? Answer is no. C1 just touch the active antigen antibody complex and becomes activated. And once it is activated, it produces the hydrolysis of C4 and then C2. So C4 break down to A and B component, B remain bound, A diffuses away. A diffuses away, A from away, going away, right? Then C4B will activate C2B.